Okay, so in this lecture, I'm going to pick up where I left off, which was finishing up with the glomerulus and the various cells that control glomerular function or the formation of filtrate. And just as a quick review as far as important capillaries and vessels or nephron structures, remember we have a glomerulus here with a glomerular capillary inside. The Bowman's capsule is another name for this structure. This is where filtrate is formed. That filtrate then enters the tubular structures. The first part is called the proximal convoluted tubule. Next up we have the descending limb, the loop, and the ascending limb of the loop of Henle. And you may have to know those in order, not just loop of Henle, but descending and ascending. Descend before you ascend. From the descending limb of the loop of Henle, we go into the distal convoluted tubule and then into a collecting duct, where this filtrate then is referred to as urine. The reality is the filtrate in the collecting duct becomes urine, but for test purposes, you're going to call it urine if we're in a collecting duct, which means this would be the microscopic structure where you first hear about the term urine. All the parts of this nephron, we refer to the substance as filtrate. Remember, filtrate was formed when you forced plasma at a greater pressure through a capillary that leaks. Some of that fluid leaks out. Remember, the capillary pressure in this glomerulus is higher than normal capillaries at about 55 millimeters of mercury versus your typical or your normal systemic capillary is around 18, between 18 and 25-ish millimeters of mercury. So we're forming filtrate in this glomerulus, and immediately after we enter the proximal convoluted tubule, which I've said more than once in video and in lecture, that this is the primary site of most reabsorption. Reabsorption can occur anywhere in this nephron process, as well as secretion, but the primary site, again, is the PCT. And associated with the PCT is the peritubular capillaries. And because the term reabsorption means to leave the nephron and enter the circulatory system, the best answer for where we reabsorb is the peritubular capillary, or primary site. The other vessels that you see here along this longer loop of Henle of the juxtamedullary nephrons is called the vasa recta. And the primary purpose of the vasa recta is to reclaim extra salt if needed. You should know that the outside of the cortex from the inside, known as the medulla, that the interstitial fluid surrounding the nephron is saltier. The deeper you go into a kidney, the saltier it is. Last point about the nephron before getting into some heavier fizz is that the beginning portions of the nephron are going to be more concentration pressure gradient driven mechanisms versus the distal convoluted tubule, or the back end of this nephron here, including the collecting duct, is more of a hormone driven process for filtrate, reabsorption, filtration, and ultimate urine formation. And when that filtrate leaves the glomerulus, the reason that we say the proximal convoluted tubule is the primary site of reabsorption in the nephrons is because it's got a greater surface area due to the brush border of microvilli. So next up we jump to some fizz. I do want to remind you that when we say renal blood is clean when it leaves the veins, I mean it's been cleansed of nitrogenous waste. You should remember a liver and kidney relationship, and that was the liver through metabolism forms urea. Urea was a nitrogenous waste product formed from amino acids. When we say renal vein blood is clean, we mean it's been cleansed of nitrogenous waste. So what you'd remember is that Again, urea is formed by the liver and eliminated by the kidneys. When we work our way through the nephron, there's going to be 3 steps that you need to keep straight of processes occurring. Again, the first step is the formation of filtrate, which occurs in the glomerulus. Filtrate formation, once formed, passes through the various tubular structures, the remaining portion of the nephron. Remember, you had different groups of cells that were important and relevant to the formation of filtrate. You had an incoming arterial and an outgoing arterial. You notice the difference in size or diameter of the incoming and outgoing, which would contribute to that greater pressure in that filtrate formation. You should know the kidneys produce roughly 125 milliliters of filtrate per minute, or if we go to the daily total, it would be about 180 liters a day. And 99% of what gets filtered gets reabsorbed. And you think to what's the normal urine output in a human, we're looking at somewhere between 1 and 2 liters. Obviously that depends on consumption. So back to the 3 steps. First we form filtrate, then we're going to have the reabsorption of materials back into the bloodstream, and if there's any metabolites or excess solutes within the circulatory system, we secrete that into the nephron, all of which is regulated by hormones and nerves, which we'll talk about here shortly. So we look at this glomerulus and we look more carefully at the type of capillary within. You can see that we've got fenestrated capillaries, which were the middle as far as 3 types of continuous, fenestrated, and sinusoidal, left to right as I said them, or first to last. The sinusoidal leaks the most and is more common, say, in a liver, or glands that might produce large hormones that need to enter circulation. Your fenestrated are your typical capillary of kidneys. On the outside you can see that podocyte, which was mentioned in the previous lecture. Again, the podocytes have feet, thus the word foot cell, and those feet move closer together to limit what can come out of the pores. Together, that epithelium in the fenestrated capillary and the podocyte on the outside of the structure contribute to what we call a filtration membrane. And again, if something is big enough to pass through this opening or pore, it enters and becomes filtrate, enters the glomerular capsule. Again, these feet of the podocyte can move closer together or further apart, allowing material to come through. And if, again, a large enough substance gets hung up here, through enzymatic action, we can degrade that macromolecule should it enter the filtrate and get hung up. Again, keep in mind what's driving this is hydrostatic pressure. The fact that we have greater pressure out here in this capillary than out in this space. Again, the pressure being about 55 millimeters of mercury. Now, what passes through this pore? Again, anything small enough to fit through, such as glucose, amino acids, some nitrogenous waste, such as urea and water, and many other things, but those are the things that you would think of and spend most of your time thinking about. Now, this process occurring in the glomerulus is known as glomerular filtration, and there's a rate of filtration, which I've said already, which is 125 milliliters per minute. Again, that filtrate, once it enters the nephron structures, is going to be regulated, again, initially by just following pressure concentration gradient. But when we get out in the distal structures of the nephron, I had mentioned that we have control mechanisms. Again, the podocyte would be a form of regulation. It would be an intrinsic factor because it's within the nephron, versus when we talk about extrinsic control factors, we would talk about a sympathetic nervous system, because your sympathetic nervous system responds to stress. Stress causes elevated blood pressure, elevated blood pressure relates to the whole renin-angiotensin mechanism, which you would be expected to know for your next examination, which is over urinary and fluids. So sympathetic point real quick, you should remember the two primary substances were epinephrine and norepinephrine. Epinephrine impacting that juxtaglomerular apparatus, stimulating the release of renin by the granular cells, also known as juxtaglomerular cells. The renin-angiotensin mechanism I'll explain here shortly, but first I would also remind you that norepinephrine was the other substance involved. And as far as systemic circulation goes, norepinephrine has an effect of causing vasoconstriction to blood vessels. And so what you see on the screen here is the primary mechanism related to renin. I would expect that you remember that angiotensinogen was an inactive plasma protein that was produced by your liver. It's activated by renin, which was produced by the juxtaglomerular cells of your kidney into angiotensin 1. Once activated to 1, ACE, which stands for angiotensin converting enzyme, activates 1 into 2. ACE was produced by your lungs. And angiotensin 2 has a long series of effects in the body. And one by one, if you don't look ahead, we stimulate the adrenal cortex to produce aldosterone. You should remember aldosterone was a steroid hormone produced from cholesterol, or a cholesterol steroid derived hormone. Again, its primary effect is to affect sodium levels. Sodium reabsorption, meaning going from the tubule back into the bloodstream. Remember, wherever sodium goes, water wants to follow if it can, and it can throughout the nephron and collecting duct, except for the ascending limb of the loop of Henle, where water is impermeable. It also stimulates the release of ADH from the hypothalamus. Remember, the hypothalamus produces vasopressin, also known as ADH, and ADH directly affects water reabsorption by acting on the distal convoluted tubule and collecting duct, affecting the permeability of cell membranes and water pores, or aquaporins. Remember, ADH also can, by its other name, vasopressin, cause constriction of blood vessels, which would also contribute to blood pressure. So those first two are affecting sodium and water, and if you affect sodium and water, you're going to increase volume in the bloodstream, and by increasing blood volume, you increase blood pressure. Next up, you got the stimulation of the thirst center, which is located in the hypothalamus. Hypothalamus making you drink when thirsty, or when prompted by dry mucosa in the mouth. And lastly, there you can see that angiotensin, by its name, stimulates constriction of arterial walls, specifically the smooth muscle, which is known as the tunica media when we're looking at anatomy of a blood vessel. So that's a long cascade to keep straight. Renin increases blood pressure, and it is a negative feedback mechanism of when you have low blood pressure, you stimulate the release of renin, and that came from that cuff of mechanoreceptor type cells known as granular cells found along the afferent arteriole. We talk about reabsorption and secretion. Again, your kidneys can clean you out of fluid very quickly within about 20 to 30 minutes. You should notice when you study this figure on your own that you've got blue and green squares or rectangles, implying that green would be secretion. You're going from a bloodstream into the tubule, while blue is from the tubule out to the blood vessel, and that would be reabsorption. So what you might wisely do is know what is reabsorbed and know what is secreted and where, by working through the proximal, the descending limb, the loop, the ascending limb, the distal convoluted tubule, and collecting duct. Remember, you form about 180 liters of filtrate daily, and you reabsorb 178 or 179 of those liters, which is about 99%. Most of the energy that's used is going to be used to move sodium. And when we talk about active transport, you should recall that that implies you need ATP, because ATP is involved in moving materials against their concentration gradients. Active transport requires ATP. Passive transport processes involve concentration gradients, concentration and pressure gradients. Material such as solutes goes from high concentration to low concentration, and water is attracted to solute load. So water goes up concentration gradients or towards the saltier side, again, if it can. What's reabsorbed in the proximal convoluted tubule? We said most things, that 99% of all reabsorption occurred between the PCT and the peritubular capillaries. All of the glucose, which would have been located in that glomerulus filtrate, all of the amino acids, most of the sodium, water, and bicarb. Within the loop of Henle, I mentioned to you that we have an ascending limb which is impermeable to water. Other hormones that can act on the distal convoluted tubule and collecting duct would include the hormones already actually mentioned, because they were part of that renin-angiotensin cascade, and then eventual stimulation of adrenal cortex and hypothalamus. But notice at the very bottom we're looking at the antagonist hormone to that entire mechanism, which is known as atrial natriuretic peptide. Atrial natriuretic peptide, by its name, tells you that your heart produces a protein-like hormone that causes you to pass salty urine. And what I'd have you notice here is that we're not saying that we're moving sodium from the bloodstream into the tubule. We're saying that the sodium that is in the filtrate, remember filtrate was formed in the glomerulus, you would have forced sodium, water, and various other electrolytes like sugar and other cations and anions into the nephron. Well, what AMP does is it prevents sodium from being reabsorbed, which means it remains as filtrate and then is passed through the active micturition in urine. So aldosterone does affect the reabsorption or increases the permeability and reabsorption of sodium. AMP prevents the reabsorption of sodium specifically. Remember, urea was a nitrogenous waste product produced by your liver and eliminated by your kidneys. We're also going to eliminate metabolites from digestion, as well as breaking down drugs that we've consumed. But not all drugs are actually broken down completely. You can find pharmaceuticals in urine in the water supply. Birth control and some of your antidepressants actually are two known pollutants in wastewater treatment facilities and found in the environment, affecting animals such as fish and amphibians. The tubular secretion process would be increasing elimination of potassium, and that would have been due to aldosterone, as well as affecting the pH of your urine by affecting cations such as hydrogen. I ask that you look up the definitions of osmolality, that you read on your own specifically about renal clearance, about glomerular filtration, and the standard substance used for renal clearance known as inulin. Again, inulin is a substance that's not reabsorbed, stored, or secreted, which means once passed in filtrate, it stays in filtrate and gives us an idea of how long and how efficient the kidneys are at clearing the substance from the body or from the circulatory system. Remember, we discussed earlier the importance of pressure, and that pressure was higher within glomerular capillaries. You can see two blue arrows which with an HP on it, indicating that this is water or hydrostatic pressure. You have 55 millimeters of mercury pushing outward and 15 pushing in. The two would be subtracted from one another. And we'd also include in our math here, we would add these two numbers together and subtract from 55 to come up with a net filtration pressure of 10 millimeters of mercury outward. Now, if someone had hypertension, that would obviously increase this number, or if someone had low plasma protein such as albumin in their bloodstream, that would reduce this number. So there are a lot of factors, natural and unnatural or disease processes, that can affect GFR. Again, efficient kidneys produce about 1 to 2 liters of urine daily. And there's a difference in the chemistry of urine. You notice the arrows pointing out the osmolality of urine. On the left side, we're looking at dilute urine with a milliosmol content of 100, versus in the concentrated scenario or case, we're looking at 1200 milliosmols. And what that means is we have a urine that is hypoosmotic, or contains a lot of water and very little solute load or concentration, while the concentrated urine is referred to as hyperosmotic and contains a heavy load of solutes. Notice the primary hormone in both cases is ADH. When ADH is lacking, we form a dilute urine. We pass more of that filtrate, essentially. Remember, the purpose of ADH was to reabsorb water from the filtrate. ADH inhibits the process of diuresis and limits how much we urinate in a day. What remains in this lecture then is to glance at and review gross anatomy, which would be the ureters, the bladder, the urethra, and understand urinalysis related information. Lab studies. Remember, the ureters were collective tubes. They collect urine from the renal pelvis and transfer that urine to the urinary bladder through peristaltic action of the smooth muscle. Remember, ureters, bladder, and urethra would be considered organs because they have 2 or more tissues. The inner lining of ureters would be mucosa, and again, the muscle of the wall would be smooth muscle. Remember, the ureter and the urethral openings are at the bottom of the bladder in an area known as the trigone. When we look at a bladder, you should recall that it has epithelium on its inside, mucosa, and that epithelium is transitional. And you should remember when you studied chapter 4 that meant that there were different shapes of epithelial cells that permit distension of the organ. Muscles found at the inferior aspect of the bladder would include the detrusor muscle, and when we urinate, we would relax that muscle. You get the urge to urinate with every 200 milliliters of urine occupying your bladder, and that urge is felt in your cerebral cortex in the insula, that deep central lobe, if you recall, located in the lateral sulcus. The urethra is a discharge muscular tube lined with mucous membranes in both sexes, about 2 inches long in females and 8 to 9 inches in males. You'd remember the male anatomy that we had 3 different terms for the urethra. We had prostatic, we have membranous, and we have spongy. And the spongy is located in the corpus spongiosum of the male erectile tissue. The trigone, again, is the structure, triangular structure found at the bottom of the bladder, where you have 3 openings. As far as urine goes, remember that the color of urine is due to urochrome. It can be affected by the foods that you eat and the drugs or vitamins that you might consume. Remember, urochrome, which is the primary pigment of urine, is derived from bilirubin, which is derived from hemoglobin. As far as the smell of urine, it's slightly aromatic, meaning it has a mild odor. But if it stands for long periods of time or is rich in ammonia, that can affect the smell, as can ketones, which are byproducts of unhealthy physiology, such as diabetic. Certain vegetables that we consume can change the smell of urine, as well as your own health status and diet. Urine has a pH that is wide in range from acidic to alkaline, but normally we say it's about a 6 on a pH scale. Depending on your diet or metabolic health, again, that pH can vary. Specific gravity was a term for how heavy is your urine. There is no unit of measurement. It's just a range of numbers from 1.001 to 1.035, 1.035 being heavier in solute load. Remember, urine should be free of glucose and protein and amino acids because they should have been reabsorbed in the proximal convoluted tubule. Remember, water is the most abundant substance of urine, and second most abundant substance, making up about 2 to 3% of that solute load, would be the substance known as urea. Again, the average person urinates about 1 to 2 liters daily, again, but that depends on consumption. Urea is a nitrogenous waste product that was produced by your liver through the process known as deamination. When you strip an amine from an amino acid, you're left with a keto acid, a metabolite that could enter the Krebs cycle. Creatinine is another substance that's used to do a rough estimate of renal clearance and efficiency. Creatinine is a metabolite of creatine phosphate, which is from skeletal muscle. Uric acid is a byproduct of nucleic acid metabolism, meaning DNA and RNA. BUN, or blood urine nitrogen studies, blood urea nitrogen studies, is used to measure damage and efficiency. And you should also, at this point, I would remind you, review renal failure and what does that mean. And there's chronic and acute renal failures that you should be aware of. Micturition is a process that's controlled and regulated by your pons. The pons, you remember, was one of the primary centers for panting and peeing, or micturition. So controlling the micturition effort is a pons function, but sensing a full bladder would be the insula. Reflexive urination occurs. That's a process from 2 to 4 years of age until a person learns to control that skeletal muscle sphincter located at the base of their bladder. The external sphincter is the voluntary or skeletal muscular sphincter. As far as urinalysis goes, would expect that you know that there are substances that we measure in urine, substances that are normal, and substances that are considered abnormal. Would expect that you would review urobilinogen, ketones, sugar, proteins. There is a urinalysis abnormal table at the end of the urinary chapter, as well as a appendix at the back of the book that talks about normal and abnormal urinalysis. This concludes your urinary lecture, part 2.